Good afternoon, everyone. My name is Harender, and I am a PMR fellow from Department of Biological Science and Bioengineering at IIT Kanpur. So this is a lecture series of class 12th biology. That is the ninth chapter, Biotechnology Principles and Processes. In this lecture series, till now we have covered the topics that are related to the principle behind the biotechnology, like biotechnology. And in the part two session, we have been trying to understand what are the processes that have been involved in the biotechnology. Uh, in this biotechnology, a particular part of biotechnology that is RDNA technology specifically. So we have been discussing about what are the processes that are involved in RDNA technology. So let me give a brief recap of what we learned the last session. So in that last session, the one of the most important tool that we learned was PCR, that is polymerase chain reaction. And for this, Carey Mullis was awarded for this particular process. Nobel Prize was awarded. So that is how impactful is this process as such. The Nobel Prize to Carey Mullis. So this scientist was able to figure out a method where we can amplify the DNA outside a cell. So this PCR is also termed as cell-free amplification of gene of interest. Till now in the part one session, you might have understood that in order to mass amplify a gene of interest, what we did was basically because this is the gene and we cloned it in a vector. Then this vector be transformed into a bacteria, mostly E. coli, has the doubling time which has the doubling time of 20 minutes. So every 20 minutes, the cells divide, and along with the cell, the plasmid containing a gene of interest will also divide. And then after when we culture this in a tube, basically, after 14 to 16 hours, we have to break all the cells. Like I hope you remember how we break cells for the bacteria. It was all one of the compound was lysozyme, which was able to break the cell wall of the bacteria, and then we were able to isolate the DNA. Similarly, we can use lysozyme to break the DNA, break the bacteria, and then isolate this, break the cell, and then isolate the plasmid. And this is like a very big, lengthy, and as well as tedious process. You are for example, you are spending time in overnight culture, and then you need to make a lot of buffers to break open this, to check the purity of the plasmid. The purity of the plasmid is very important, and then you also need to check what if there is contamination and everything. So, this is a very long process, very tedious. So, this method as a whole. This method as a whole is cell-based, cell-based because we are using a cell, a living organism, to amplify our gene of interest. So this is our cell-based amplification. So as you could understand that how long it takes and what are the disadvantages of this. So this method was we still use this method during the cloning process, but when we want to isolate the only gene of interest, we want to mass amplify the gene of interest, we can just do PCR, and the PCR within 2 to 3 hours you will get a better yield of your gene of interest. That's why this cell-free amplification method, that is the PCR, has gained a tremendous limelight in the field of us, the field of biology. And since this PCR is so impactful, therefore, uh, the scientist, the scientist itself was awarded with the Nobel Prize, the highest prize get in biology. So we were learning about PCR, right? So PCR, we understand that it is polymerase chain reaction. So I hope you remember these major steps of PCR. This denaturation, then we had aniline, and the third one was extension. So in a basic term, I can tell you that denaturation is basically breaking of the DNA bond, breaks the hydrogen bond between DNA strands. Aniline is basically uh, the prime. It is stage where the primers anneal to the complementary sequence. Third, extension. Here the DNA polymerase acts up. DNA polymerase. The In the case of PCR, we use special type, that is stacked DNA polymerase, because it is very stable at higher temperature. So DNA polymerase we use to amplify our gene. So this is the basic role, basic steps of a PCR. So visually, if I draw it for you, like for example, you gave it a DNA sequence, right? You gave a DNA sequence in the PCR, and then due to denaturation, that is at 95 degrees, denaturation occurs, and then you will have a single strand, something like this. So we are starting with one, right? Now we have the step of annealing. Annealing, it is at 55 degrees. So in the annealing step, here's the DNA. In the annealing step, the primers bind. This is the first primer, and the second primer. Now, then we have the extension step, wherein the polymerase comes in. Extension, that is at 72 degrees, and here polymerase comes in, and then it basically amplifies the entire sequence. So it is something like this. We have two strands, and then on the two strands, based on the complementary sequence, the primers bind. And when the primers bind, so with the action of polymerase, the tag polymerase, the strand will amplify, extend basically. So as you started with one strand, the yellow color that I'm using is just for your reference. There's nothing scientific about it. So after this one cycle, as you can see from one, this is 2 DNA. Therefore, if you want to calculate number of DNA after n number of cycles, n cycles, the formula is 2 to the power of n. For example, in this case, there is only one cycle occurring. So 2 to the power of 1 is 2. Basically, at the end of the day, you got 2. What if there are 4 cycles and 2 to the power of 4 is for the 8, 64 cycles, 64 basically. So at the end of the 4 cycles, it will be 4. And normally we run around 25 to 30 cycles. And you can understand like how much DNA will get at the end of the PCR product. So that is it. So that's why it is very important and very powerful technique in the field of biology. So I hope you have understood. So this was the brief of the last session. And this is really very important, one of the most important techniques that we use on a day-to-day basis. So moving on, just remember this circuit. So there are some points to remember here. Points to remember. First, remember the formula, number of the yield. The formula gave us point to remember. Yield of DNA stands after yield of DNA stands after n cycles. So you should remember this formula, very important formula. This to the power of n. Simplest formula, but very important. Then you should remember the PCR, the steps, denaturation. You just have to memorize this. There's no other option. Once you by heart this, it will be easy for you to understand what is the entire process. So and the temperatures as well, 95, 55, and 72. So this and this, like, and denaturation in annealing has a range of temperatures, but you don't need to get into that. So it all depends upon your desired size of the pad and everything. So you just remember three temperatures: 95, 55, and 72. But just for your reference, it goes from 95 to it goes from 94 to 98 degrees, and this can go to 55 to 70 degrees. It depends on the primer. And this is 100. But you don't need, so there is a range for each step. So this is the thing. Now moving on. So we have already amplified our gene of interest. We have our vector digestion and everything done. Now the next step is insertion of recombinant DNA to host cell. So what is recombinant DNA or the recombinant plasmid? We have a plasmid normal vector and in which our gene of interest is cloned. That is recombinant DNA because it's a chimera. They both are not. They do not occur naturally together. So we have made a chimeric DNA or the recombinant DNA. So using any methods, the ligated DNA produced to recipient cells. They take up the DNA from its surroundings. So we have learned about one major common method, that is transformation. Transformation. And I hope you guys remember what transformation is. It is basically a heat shock method. So at the 42 degree, we give a heat shock. Normally the bacteria, the competent cells are high. After the DNA is added, we give it a heat shock at 42 degree, which opens up pores in the DNA, the cell, and the host takes up the DNA. That's what they are saying. Using any method, in our it's a heat shock method, that is transformation. The ligated DNA is produced in the host cell. So we use the heat shock method. there is electroporation method. Again, there is also transformation. So yeah, so transformation as such is defined as a process by which a host takes up a naked DNA from the surrounding. Now, this can be done by heat shock method, electroporation, then there are like other condensation methods and everything. So you don't have to memorize anything. At your level, you just have to remember the transformation occurs, and we use heat shock method. Now, if the recombinant DNA bearing ampicillin resistant gene transferred into E. coli cells, the host cells become resistant. So I hope like one thing you should remember, or like I have already taught you about this, that we use a competent cells. Competent cells. Now there's this normal cells. This like the name of the competent cells that we use is DH5 alpha, and in this DH5 alpha, this clone genome, there is no resistance. DH5 alpha has no resistance, so it will die on any antibiotic plate. So you can grow it on ampicillin, you can grow it kanamycin, you can grow it tetracycline. It will die. It will die. But now, after transformation, when you put up a plasmid, like let's say this is your plasmid, and this is a gene of interest, and in this gene of interest, there is this ampicillin resistance. So once this bacteria, again, the same DH5 alpha, once it takes up the bacteria, sorry, plasmid, and now since this plasmid has the ampicillin gene, it can grow on ampicillin, on ampicillin plates. This is straightforward, right? So that's what if a recombinant DNA bearing ampicillin resistant gene is transferred into E. coli cells, the host cells become ampicillin resistant. That's why they can grow easily on the ampicillin plate. Now, if the transformed cells are spread on agar plate containing ampicillin, only transformants will grow and recipient cells will die. So as I already mentioned, that we have this DH5 alpha. Like what if this DH5 alpha takes up the plasmid, the ampicillin plasmid, and this doesn't. So this will die and this will survive. And that's how we'll get the colony containing the ampicillin with the transform. So here's a better representation that we have this recombinant DNA, we have this E. coli, and then we transform it. Now, if we transform, some cells will take up the plasmid, some won't, right? Some won't. So once we grow this on a plate, these cells will die. They do not have any resistance against the antibiotic. Antibiotic will kill this non-transformed cells, and this will survive. That will form a colony. This is it. Next, now the fifth part, that is obtaining the foreign gene product. Obtaining the foreign gene product. Now, the ultimate aim of recombinant DNA technology is to produce a desired protein. We have already understood this concept that, for example, I most often give an example of insulin. That insulin is a protein that is very important for diabetic people. And if there is a protein, there must be RNA. If there is RNA, there must be DNA. That means the gene level. So the ultimate of the recombinant DNA technology is to produce a desirable protein. So our desirable protein is insulin. So if a protein coding foreign gene is expressed in a heterologous host, it is called a recombinant protein. So till now we were studying about recombinant DNA because we knew that the backbone is of different origin and the gene is of different origin, right? They are not naturally occurring. Similarly, if the gene is put into a host and it expresses a protein, and the protein encoding one and the protein expresses the heterologous host, it is a recombinant protein. So that's a straightforward answer. So what is recombinant DNA? It is basically recombinant DNA is basically a plasmid molecule or the vector molecule containing a gene of interest, which is not basically naturally occurring. If I tell you in the simple terms. Similarly, a recombinant protein is a protein that is encoded in a heterologous host. Heterologous host means that the protein is not natively expressed in that particular host, and therefore it is a recombinant protein. Now, cells with foreign genes can be grown in laboratory. The cultures are used to extract the desired protein and purify it using a separation technique. Now, this is very important and very advanced thing that in the I think in the chapter 8 you might have understood of how we can grow bacteria or fungus or algae or any kind of thing, any even viruses in laboratory. So similarly, the host that contains a gene of interest can be grown in labs. And once this grow, and if the cell has the machinery to express this protein, we will have the protein, and then we have to isolate it. So can be grown in labs, and then uh, isolation of the protein by separation methods. Now, what are these separation methods? So this again is a very wide topic, but I think you might have heard about chromatography, not the paper chromatography that you might have heard in your biology classes. There are like multiple advanced chromatography techniques that are uh, very important to purify a gene of interest. Like for example, I can tell you affinity chromatography. Then there is nickel-based chromatography, then size exclusion chromatography. So every other chromatography has its own property of why it is used, and based on what our desired product is, we have to use different chromatography techniques. So once we transform a gene of interest into a cell, and if the cell is suitable enough to express our protein, then we have to again lyse the cell as well as uh, lyse the cells, and then purify our protein and by a separation method, that is chromatography. Now, so this is done. So this is called chromatography. Now, the cells can also be multiplied in a continuous culture system. Here, the used medium is drained out from the cells can be multiplied in a continuous culture medium. Here, the used medium drain out from one side while the fresh medium is added from other. It maintains the cells more physiologically active, so produces a larger biomass, yields more desired protein. Now again, uh, there are like various culture systems. Uh, what for example, what we have is we have the bacteria with our gene of interest, and we know that this bacteria expresses a protein, expresses the gene to make the protein. Right now, this now we have to mass culture this bacteria, because every bacteria will produce certain amount of protein. So the more number we have, the better will be the yield. So in order to culture this, there are like two most prominent methods. One is continuous culture system. And in this continuous culture system, what happens is, let me tell you clearly. Then let's say there is this, let's say a container, wherein we'll add all the we'll add the media, we'll have the media, then we will add the media is for the growth. Second, we'll add the cells, the bacteria that has our gene of interest, and then we'll basically culture them. And in the continuous method, what happens here is that again, since the bacteria, we have to keep it for a longer period of time. So we will grow this bacteria for particularly, like let's say, depends on the type of protein that we want, and then we let it to grow. And after a certain period of time, again, since the bacteria is utilizing all the nutrients in the media, then obviously the nutrients are basically getting exhausted. So for example, put 1 gram of glucose, they will utilize it for their own use, then there won't be any gram of glucose. So what we do in this continuous system is that we will remove liquid media slowly, and then we'll add a fresh media. Remove exhausted media. Let's use the word exhausted media that is used up, and then add fresh media. And therefore, we do not need to stop this culture. We can go continuously, and in this culture, we'll get the yield, best and more high yield. Another method that we have is batch culture system. What will happen? This is we have this normal, let's say, like use flask, and then we add all the bacteria, we add the media, and then let it grow, media and bacteria. And once 40 to 6 months later, we just culture it directly. So the yield here is just taken out. So yield here is low. So that is the difference. In the continuous culture system, there is a continuous addition of fresh media as the removal of exhausted media. But in the batch culture system, there is a stagnant one basically. It is like one time addition and then one time completion. This is in this the yield is quite low, because as I can compare, like in the batch system, if I give 1 gram of glucose, it will utilize, let's say, 1000 bacteria grew. Here again, I also gave 1 gram glucose in the first go, 1000 grew. But again, when I removed the exhaust and added 1 more gram of glucose, this became 10 to the power of 6, double. So that's how the difference is. So that is what the chapter talks about, that the cells can also be multiplied in a continuous cell system where the use of media is drained out from one side while the fresh media is added from another. And it maintains the cells more physiologically active, so produces a larger biomass, and it eats more desired protein. That's what we have done. Now, again, the concept of bioreactors was the same thing that we learned. Uh, is the same thing that we learned in chapter 8. So again, now to obtain the foreign gene product, we have to use a specialized vessels, bioreactors. So what are bioreactors? So these are the vessels in which raw materials are biologically converted into specific products, enzymes, etc., using microbial, plant, animal, or human cells. So what are bioreactors? They are basically vessels used to produce a desired product at a larger scale. So this, like what we do is basically this vessel, you can see here, they are very, like I think you have, the properties of this bioreactor are very specialized. They maintain the temperature, the humidity, the aeration, everything. And they have all the parameters to look at the pH and everything. That is very important for the sustained growth. Because if this bioreactors or the bioreactors are used at an industrial scale, and this are helpful in producing product at mass, like they are helpful in mass production of product. They're not used at a smaller scale. So upon addition of this raw material, they are converted into products by the use of the by the use of the bacteria or any living organism. The raw materials are converted to specific products. And then now these bioreactors are used to produce large quantities of products. These bioreactors are used to produce large quantities of products. They can process about 100 to 1000 liters of culture. Again, so I hope you are able to grasp the amount, massiveness of these bioreactors, that they can hold up to 1000 liters of culture. And and therefore it has to be maintained at a very, very crucial condition, better condition, so that there is no contamination, there is a proper growth phase, proper nutrient supply, proper pH, proper temperature, proper mixing, and everything, so that we get an optimized and a very good yield. Now again, this bioreactors provide optimal growth conditions, this temperature, pH, substrate, salts, vitamins, oxygen to get desired product. So whatever, like first of all, it should be sterile. We have already talked about this. That sterility is very important because there are multiple microbes and everything that are living among us. So in order to just grow the specific bacteria or the one that is very useful to us, we need to have a sterile condition the in the bioreactor. Now this bioreactors maintain an optimal growth condition. So it is depending on the type of organism that we use, what kind of conditions they require. Uh, that is very important. Like for example, if there is a bacteria, the pH and the temperature will be different. Yeast or the fungus, the temperature and the pH will be different. Similarly for the animal cells or the cancer cell line, anything. And therefore, and again, even in the depending on the type of product, if you want to make enzymes, want to make antibodies or vaccines, anything, that everything is like very conditioned, very specific to their uh, own part. Now, the most commonly used bioreactors are of stirring tank, stirred tank reactors. So I hope you have like we have already discussed. So let me tell you about this thing again. First, let me show you with this diagram itself. What we need is a stir type reactor. So stir. I hope you understand stirring basically mixing type. So that why do we do that? So that the culture mixes properly and the nutrition is and the nutrition given to all the uh, it is available to all the bacteria. Like what are the organisms present in the bioreactor? The nutrient is available to them. So it is It is usually cylindrical or with a curved wheel, facilitate the mixing of the reactive content. So we have this cylindrical and we have the curve. It is basically a cylindrical curve based uh, reactor. Why it is very important? So that it helps in the mixing of the reactive content. Now the stirrer facilitates even mixing and oxygen availability. So this we have the again we have the central fast and it is basic the stirrer uh, and it helps it even mixing and oxygen availability to the living organism over the cells in the bioreactor. Alternatively, air can be bubbled through the reactor. So this one is a simple stir tank bioreactor that is cylindrical and it's made up of stainless steel and everything we have already understood. Then there is for the growth of the optimized growth of the living organism. We have this acid base for the pH control. Then we have steam for sterilization to remove all the contaminants. Then we have this motor to rotate this fast. Uh, there is a stirrer, and then we have foam breaker. Then we have impellers. Again, these blades are called impellers to create the thing. Then we have the culture broth, and then there is this air, right? So this is the common stir tank reactor. Simple. Then we have this sparse stir tank bioreactor, through which the sterile air bubbles are sparse. So as you can see, this is a very simple design, but sometimes it is this kind of simple force on the reactor is needed by uh, by the organism or by the product itself. So in the sparse one, what we have is there is a sparser here called sparser. So what is sparser basically? Just like your shower basically, it is a these are the nozzles, and from this below this, it will give up the it will give in a sterile air. So this sparger will give sterile air. So this sterile air, and then we have this motor connected a rotor as well, motor as well, and this will spin. So this again this sparger and this motorized mixer is also helpful in mixing of the content. So uh, the bubbles dramatically increase the oxygen transfer area, and then that surface area for the oxygen, and there is a proper mixing of it. So we have learned about two different bioreactors. One is stir tank bioreactors. Another is sparge stir tank. So in what is the difference? Sparge one, there is this sparger present that will basically release air in the media. Now, what are the parts of a bioreactor? So what are the parts of the bioreactor? We have an agitator system. Agitator system. So what is the agitator system? Here, like uh, in the agitator system, we have this impeller. So in the agitator system, we have impellers. Then we have motor, and we have this foam breaker. And in this, we have sparger. So in the agitator system, we will have the motor, impellers, and sparger as well. Then we have an oxygen delivery system, depending on the bacteria, depending on the type of the organism that we use, whether it is aerobic or anaerobic, we can supply the oxygen. Next, we have a foam control system, so can lead to frothing. So we have, like for example, you might have heard or seen, in your kitchen or like a mom's kitchen that once like there is a lot of bubbles or there is frothing, if we just add vegetable oil on top of it, the foams will go off. Similarly, in this bioreactors, we have foam control system. Next, we have our temperature control system. In order to maintain the temperature, like for example, for the bacteria, 37 degrees is very important. Um, for bacteria, 37 degrees for optimal growth, but for fungus, the yeast is 30 degrees. So that's why temperature control is very important depending on the type of organism we use. Then we have the pH control system. So there are like certain things, certain proteins that express the lower pH, the optimum pH, and there are higher pH. So pH system is also important. Then we have the sampling port for the periodic withdrawal of the culture. So again, sampling port, as the name suggests, people usually take some amount of the sample and check of the contamination and everything in order to. That's why the sampling port is important to test the what are the raw materials that form, what are the products that form from the raw materials, is it right or not? So now, now there is this downstream process. So this again is very important. So till now, what we have learned that we started with the gene of interest. We have this insulin gene. Now we clone it into a vector. Now the insulin gene is in a vector. So we have all used all the restrictions. We have isolated, we have therapied everything, and we have cloned it. Then we have put this thing in a bacteria. Then we do a primary culture to get enough numbers, and then we go over a secondary culture. In the secondary culture, where there is a very optimized growth, protein expression is done. Protein expression is done. So the primary culture is just to increase the cell number, but the secondary culture is for the protein expression. So we have this, and the secondary culture. Let's say we have this bioreactor. So after the secondary culture, we need to let's say there are like the product. That is the product form. Okay. So the product, we have the product now. We know that we have a product now. Where is the product? Is it in the media? Is it inside the cell, or where? So depending on where the product is formed, we have to use different methods of purification methods to isolate the protein. In order to isolate the protein. So if the product is formed is released in the media, for example, there are certain bacteria that are they make the protein and release in the media itself. Then we have to collect the media and then process it accordingly. Then if the product is found inside the cell itself, then we have to isolate the cell, pellet it, then we have to break open the cell in such a way that it does not harm our product. So again, the down process will occur. So that's what downstreaming process is. It is a series of processes such as separation and purification of products after the biosynthetic stage. So till now we have cultured them. We have known that the product is formed either in the cell or in the media. So based on that, we'll isolate either media or the cell. So this down processing down. Then after isolation of that, we'll start our downstream process. So this is a series of processes such as separation and purification of product after the biosynthetic stage. So this process basically helps us in order to give us a purified and better product. The product is formulated with suitable preservatives. Such formulation undergoes clinical trials and strict quality control testing. So since after this process, the products will be supplied to living organisms such as us or even any animals as such, that is animal feed. Uh, to before giving them, there are like first the product has to be preserved, whether because obviously there might be a shelf life of any protein or anything. For example, insulin is usually stored in at 4 degrees inside the fridge because it is not that stable for long in the only at the room temperature. So again, you should remember that when I'm telling you that the product formed inside the cell at 37, it is just the machinery from the RNA to protein, the translation. But after the protein is extracted from the cell, it isn't stable. It won't stay long at 37 degree, and it will degrade. Therefore, the story is always 20 minus 80 of 4 degrees. So these are the processes. Uh, so upstream process till now we have understood that there is cell number, oxygen, time, RPM, and after everything, we get the product. And then the downstream process occurs wherein there is a removal of all the media, all the like anything that we do not desire is basically contaminant. So, for example, if I wanted the insulin protein, then except insulin protein, everything I'll label it as a contaminant. So, for example, after this, there is pressure of surface size, response press, where the media is separated, and then we have this processing folding, then we have this purification columns again. As I told, it's a chromatography based process where based on the size of the protein, like for example, the bacteria produces a lot of protein, but I know that insulin is of 30, 36, insulin uh, is a protein of particularly 45 kilodalton. So we know that only at this size I will have this protein. And then sometimes we tag our protein with certain things. And like for example, his tag is one of the major most like his tag. It's a very important tag where we put it on a protein or desired protein and using nickel NT chromatographic column, we pull the proteins that are his tag. So I know that I've already tagged my protein with his. Uh, then this nickel NT will only will trap this his tag protein. So after this column, then we we have to consider. For example, if it is 1,000 liter culture, we will not get, obviously we won't get the 1,000 liter yield. So we have to purify after this. We have to concentrate. Then we will have the final product. Now this final product will be stored in preservatives, glycerol, or even -80 without anything. So in order to maintain the pH and the condition, and then it undergoes the clinical trials in order to approve. Okay, like for example, if I let's say there are like 10 bioreactors like this. For the each bioreactor, I'll have this unit, and from the each bioreactor, I'll have to test for the clinical trials. Strict quality control testing will be done. Even though they are the same product, even though they are isolating and doing from all the 10 reactors, each I'll have to send 10 samples, and those 10 samples will have to be tested in the clinical as well as the quality. So let me summarize today's session. And in this today's session, what we learned basically was a normal process of RNA technology. And after vector containing gene of interest is added into a host is mass amplified using bioreactors. And what are bioreactors? They're basically vessels wherein raw material material is put in, and the cells and the product is isolated. Then we learned about bioreactors. It's like we learned about two types. One is stirred tank and one is sparged tank. So this again has a cylindrical base. Again, this also is cylindrical, but there is a sparger in it. And what is a sparger? This air bubbles are basically oxygen. It helps in mixing. Then we also learned about parts of, also learned about bioreactors parts, and we learned about agitators. Example, what were the part of agitators? There were motors, then there were sparger again comes into this, then we had the impellers. I hope you guys know what it is. So basically, this is the shaft inside the bioreactor. This is the impellers. This blades are impellers, and there's motor, and we all know the sparger is like this. It will release the air bubble. Then we had the control for pH and temperature, pH and temperature controller. We have this foam controller. We had this port, sample port, sample port, yeah. And then we have this sterilization unit. And we know that the bioreactors are mostly cylindrical curve because they facilitate proper mixing of the proper mixing of the bacteria. Now, after this, after the culturing, we have this downstream process, downstream process, and they are basically steps to purify and test our product. So there are like multiple filtration steps, filtration, separation steps, and after this, that is, it is concentrated. Like the higher volume, the we get very small volume, very highly purified and concentrated protein, and then it has to go in clinical testing, etc. So after this, after we get this quality control report, QC report, we say QC report, that is quality control, then we'll have a finished final. So this is the entire process of biotechnology, the principles and all the processes that are involved in this, and how from a single gene we have we create and generate our desired proteins that are very useful to the human being. So the one of the common examples that I keep giving them, because it is like very commonly used in purchase, and you might have seen people using that. So it is a go-to protein, and uh, this is how the biotechnology field as a science is helping us in creating a better product for the mass population, and that too very easily. So this is it for today. Uh, with this, we have basically completed the entire chapter, and yeah, I hope you have understood everything from the get go. Thank you.